Okay, good morning. We will get started here. Any questions before I get into this table? Okay, so real quickly, I am nearing completing the exam. Uh, so I will have practice. Okay, good morning. We will get started here. Any questions before I get into this table? No questions. All right. Every time it does that to me. Okay. I hope I hope I'm in business now. I think I am. It looks like it's all good. Uh, okay, so I do have just a little bit left to finish with the exam. I will get a practice put together. I hope to have it up, the practice bit, up later today by the end of today, so that you've got some time, multiple days to be thinking about it. The test does not open until the 19th, so that is a bit down the road. But I think for some, uh, more time to think about it is better than less. So uh, the extra credit is also posted on there. I went over that stuff last time, but it is under the week of the 15th is where you will find that assignment. It's posted under there as a quiz, I believe, just to give you spot to type it in there. Okay, so this is out of the textbook. It is the PowerPoint slide that we left off on, but that slide was a little bit blurry when I projected up here full screen. So I just snagged this right from the text. That's what I'm sharing it out of. And this is table 713. This is a pretty good table. I have looked through it and I thought, we should spend a second just going over a little bit of it. So we've got a handful of different formed elements that we will look at as we progress through here. All right. And then the specific subtypes and then concentrations and some details about them. All right. So you don't need to get bogged down with uh, some of this stuff, like percentages. Uh, there are things I will touch on here, but we think more about some of the function. Some of these details are important to think about with these respective types of cells or fragments when we get down to the platelets. So there's a lot of things when we think about your blood. It's more than just erythrocytes or red cells. There's a lot more going on, and not that the red cells aren't important when we think about gas exchange, but when we're talking our immune response, when we're thinking about the potentials to deal with foreign invaders, to fight off disease causing agents, it's the rest of these players that we really want to think about. All right. So initially, these erythrocytes, if we're thinking about percentages, they are the main players when we think about blood. Between about 5.2 million, between 4 and 6 million erythrocytes per microliter. So truly a tremendous amount. And in a blood smear, uh, that's really, you really see the majority of those cells are erythrocytes. And then you start seeing the actual nucleated white cells. And that's, when you're doing blood counts, that's really what we're thinking about are the leukocytes. What sort of percentages do we have going on? And when we see deviations from the norm, whatever that might be, increases or decreases in those normal amounts, that raises some flags as far as potential issues that we might be seeing. And dependent upon some of the specific types of cells where we see deviations, that might give us some further clues as far as what sorts of things we might be thinking about, right? So the erythrocytes, these cells, for the most part, are the longest lived, if you will. They persist for a few months, about 120 days, give or take a little bit, within our circulation. They're constantly being formed and being replaced, but the longevity, about 120 days. The rest, as we move into the leukocytes or the white cells, as we move into these guys, it's much, much less. Typically thinking maybe a few days, maybe even just a few hours. Uh, Memory cells, when we think about B and T cells, there are capabilities of these guys being much longer lived, sticking around longer. But with a lot of these immune cells, if you will, as we start thinking about these leukocytes, their longevity is much less. All right. So again, the bulk of those cells per microliter are the erythrocytes. Then we look down to the leukocytes, and you can see now instead of talking millions, we're talking thousands. All right, and this is all of them together. All right, about 7,000 white cells per microliter. All right, ranging from 5 to 10,000. All right, and again, kind of just depends on circumstances. When we look at these, uh, the red cells are uh, lacking a nucleus. You can see them kind of dimpled in, and that sticks out when you're looking at that because they look like hollow little cells for the most part. You move down to the leukocytes, and now you can see, as I bring it up here, all sorts of different staining characteristics, but nuclei that are very apparent, typically dark purple or deep blue, and they really kind of stand out. So again, their concentrations uh, pretty low. When we move into the granulocytes, so these three players right here, uh, these guys, uh, the bulk of the leukocytes in the first place, about 4,300. The neutrophils are the biggest players, circulating through the body, and with this, these guys have the potential also to leave the circulation and move into tissues to help fight with infection as well. But again, we're talking per microliter of blood. So neutrophils, they make up about 50 to 70% of this population, right? And we oftentimes call the neutrophil a PMN cell or polymorphonuclear because their nucleus is multi-lobed and kind of odd shaped, right? As we look at it. So PMN cells, although sometimes they kind of group it and say all of these granulocytes are PMNs, really PMNs are the neutrophils. And then we talk of these guys really sort of separate, right? Kind of depends on what you're looking at with that. So again, major player are the neutrophils. They're neutral staining, and it's the granules, the lysosomes within these cells having different types of proteins or enzymes within them that pick up the staining properties a little bit differently and allows us to differentiate, right? So they stain kind of pale, sort of lilac colored. They are phagocytic cells, so they've got the capability of engulfing foreign invaders. And they're very good when we start thinking about targeting bacterial cells. They also can release granules to the outside of their cell to stimulate more of an immune response. And we'll see some of that here in just a second, right? So again, most common and their lifespan, minutes to days, depends on their circumstances. If they're actively releasing granules or phagocytizing, they don't have much time, right? And in turn, they die in the process of dealing with these foreign invaders and really, they constitute the majority of the pus that's seen in some of these types of infections. Eosinophils, these are the next ones with the granulocytes, and their percentages are much less, 1 to 3% of the total leukocytes. Typically, the eosinophils and basophils in a normal blood smear are pretty hard to find. You can see here pretty low levels, 165 or so per cells per microliter. Typically double-lobed nuclei and red-orange granules, acidic staining granules. They're also phagocytic, but a lot of times with the eosinophil, it's more extracellular killing. Instead of engulfing and pulling in that foreign cell, they'll surround it and dump digestive enzymes to the outside. They're important with some parasitic diseases, and so thinking maybe larger organisms. Things that, the neutrophil is bigger than a bacterium, but eosinophils are, well, they're smaller than most of the cells that they're going to be targeting. So they kill extracellularly instead. They associate with some different antigen-antibody complexes. And again, like I said, parasitic infection is important when we think about eosinophils. So seeing an individual with elevated levels of eosinophils is a good indication that you got something like that going on. And we start digging a little deeper into the patient histories to see maybe there is a type of parasite that we're thinking about. With that said, you also kind of need to think about where these people are coming from. If they're in an area where parasitism is relatively low, thinking maybe Idaho, not a huge concern, then we shift and think maybe it's something else. And we can see, antihistamines here as a potential that they're stimulating. Allergic reactions also can see higher levels of eosinophils. So through a little further digging, we might find out that, oh, you don't have parasites, you have hay fever, seasonal allergies. You pretty much know if you have allergies in the first place, and that's something you're going to clue the clinician in. They're going to be asking you questions like that as you go in for whatever, all right? And it may be ruled out, and deep down, there's no reason you can't have allergies and still have some parasites at the same time. That's a win-win, not for you. So lifespan also short-lived. The basophils, these are even less likely to be found, less than 1% of the population of the granulocytes, about 44 cells per microliter. Very difficult. Oftentimes looking at blood smears, you can search for a considerable amount of time, multiple fields of view as you look at these cells moving to the next bit, almost evaluating the entire slide before you might have the opportunity to find a basophil or two, right? Very low levels. Their granules are very deep staining, dark blue or purple granules, and there are a lot of them. Oftentimes, with the staining of this is a nice little cartoon picture of it, but oftentimes the granules are so prominent that it almost completely clouds over the nucleus. You can tell there's something going on there. There's a nucleus behind all of the speckling, but it almost just takes over and makes it difficult. That also makes it a little easier to kind of pick out if you're fortunate enough to find one. They're responsible with inflammatory responses, and we see some histamine with that as well. This, as far as some details about it, very uncommon, and lifespan of these guys aren't really, that's not really hashed out at this point, but likely something that's also pretty short-lived. The next group of leukocytes we've got a couple of different potentials with these guys, and we call them agranulocytes, although there are the potentials that we might see some with a few granules. For the most part, very low levels of that or non-existent, right? So these, the first bits are the lymphocytes, and as we think about these, it's more like T and B cells, which this little guy down here is maybe more what we would think about with that. And looking at these lymphocytes, you can't tell the difference between a T cell or a B cell through simple staining, right? They basically look the same. The guy that's up above it, that's got the speckles in here, this is a cytotoxic T cell. And so it's something, it's a little bit different, right? And it is something that's maybe a little easier to differentiate. And it's really, you know, it's larger, but it also has some of these granules that will lend to its killing. The lymphocytes, 20 to 40% of that, about 2,000 or so cells per microliter. And again, specifics with immunity, T and B cell wise, that we'll hit here in a bit, but thinking about stimulating these cells, some that we've already talked about and others, to come in and respond, and also the production of specific antibodies dependent upon what's being presented, are some of the roles of our lymphocytes. And with that, we have cells that are actively doing their job. When that happens, we call them plasma cells, and they start releasing their substances. And then we have memory cells that lay in wait till they're till a secondary exposure with that. So the monocytes, these guys are bigger than any of our other lymphocytes. All right, they're a little bit easier to distinguish that way when you're looking at a smear. You start seeing lots of these nucleated cells, and then all of a sudden there's one that's kind of a monster. Monocytes, definitely a little bit easier to kind of pick out that way. So big nucleus and no granules with that as well. That's a good indication of a lymphocyte of a monocyte, excuse me. With these lymphocytes right here, I also want to point out, oftentimes the nucleus almost takes up the entire cell. There's very little cytoplasm that's left over when you see the lymphocytes, right? So monocytes, big guys, they're relatively low levels within the circulation, but typically pretty easy to see, and you're definitely more likely to see some monocytes than eosinophils or basophils for that matter. So these guys have more, I mean, we see them in our circulation, but they move out of the circulation and they move into tissues and differentiate, and we start thinking about macrophages, dendritic cells, if we're talking the skin, and their job is really surveillance. They phagocytize cells, they destroy these foreign invaders. They also can, in the process of doing this, they become antigen-presenting cells, so they show proteins, they show fragments of that foreign invader, these epitopes. They show them on the outside of their cell and stimulate other types of immune cells, like showing them to T cells, which in turn can see these fragments and recognize what sorts of a response truly need to be tasked with kind of mopping up this problem. Lastly are the platelets. These guys down here are fragments. We showed last time those megakaryocytes that basically get fragmented, and it's bits of those that are the platelets. They look like very small nucleated cells, but they're really not an intact cell. It's just a fragment. They are important, and we think about wounds and platelets being some of the first substances, I want to say cells, but first substances that show up, and they have growth factors that they start to release that stimulate mitotic divisions, tell the cells, the neighboring cells, you need to kick it into overdrive. We need to start fixing this problem. And it's really the sum of the chemicals that platelets deliver that initiate this in the first place. So the hemostasis, basically keeping this stuff functioning properly by these growth factors that say repair or heal from a wound, whatever it might be. So that's the idea with those guys. Let me stop this and switch it back to this. Okay, so this basically just a blow up of the granulocytes that we introduced initially in that table to give you a real image and that kind of cartoon image that we had started with. So this first image with the neutrophil, you can see an actual microscopic image with the convoluted nucleus, the kind of stippling, the speckles within here, very pale, very light, as we said, kind of lilac colored, lavender sort of colored. The eosinophil, multi-lobed, bilobed nuclei, and very red, orange kind of staining granules. These guys, you know you see an eosinophil because everything else has kind of bluish, purplish granules if they have granules, lighter or darker, but these guys are very different, all right? They stick out. So they're hard to find, but when you do find one, you pretty much know with certainty you've got an eosinophil because it's nice and red, right? So you can see these granules sticking out. And then over here, the basophil, and you can see lots of dark granules, the nucleus, and the granules kind of right on the top of it, right? So sometimes kind of just shrouding that the littlest bit. We do have an area that's kind of faint right here, but still lots of dark granules. So they're also shown up here, the neutrophil, the greatest concentration, then eosinophil, and lastly would be the basophil as far as what you might expect to be seeing with those guys. The next one, these mast cells, they're kind of like basophils. They function similar to a basophil, and they are important with initiating an inflammatory response. When we think about the inflammation, oftentimes inflammation, you try to limit that, thinking that there are definitely issues with inflammation, but some of it, low-grade inflammation, is important because it helps to basically spur more of these cells to come in and respond. When it gets extreme, you can definitely have more of a damaging situation as a result of that. But they are important to stimulate an inflammatory response. So here you can see, here's the erythrocytes, empty little shells, if you will, no nuclei within here. And then these mast cells that are, very large, almost fuzzy looking with the granular appearance in there. So they're difficult to differentiate mast cells and basophils. They do appear very similar. But mast cells have the potential to migrate into tissues, and that's what we're seeing here, these mast cells moving in to some tissue. The basophils stay within the circulation. So slightly different. Natural killer cells, these guys also different, and they're sort of like a cytotoxic T cell, all right? Uh, but they are different than that, but they do bear some similarities to that, all right? So natural killer cells, they get inhibited by the presence of major histocompatibility complex receptors on healthy cells. So if they see, and this bit up here uh, is a normal situation, a healthy situation, I should say. So here's your own cell. It's got its MHC1. So this is our own, and we'll hit some details with the MHC complexes here in a second, but we've got it right now. Our own normal cells, nucleated cells, I should say, not normal, our own nucleated cells have a molecular fingerprint, if you will, this MHC complex, major histocompatibility complex, and in this case, type 1, all right? This is what's expressed on all of our healthy nucleated cells. You don't see it on red blood cells, and the red blood cells don't have a nucleus, all right? That's important when we think about transfusion reactions that we'll hit a little bit later. We can donate blood if you've got the proper blood type. We're not going to go into that right this second, but you can donate blood because we don't have these complexes on the surfaces of our cells. If that were the case, you would have to be an identical match to donate blood. That's an issue. And well, the Red Cross would probably be out of business. There'd be trouble. But fortunately, those cells lack it, and you're able to transmit blood from one individual to the next. There are other substances that need to match up, but you can donate, all right, without issues. So these natural killer cells are doing surveillance, and they're looking at our own normal body cells, and as long as they've got this complex, they keep on moving, all right? You're all fine and good. You're telling me you're normal, all right? But, and a lot of the time, natural killer cells, their functions really are thought with virally infected cells. It's not just this, all right? We can see some other issues with it, and potentially cancerous cells might have some variation. A lot of their discovery was with the virally infected cells and the issues that they impose upon that cell. So as this cell becomes infected with whatever type of virus, the cell becomes infected and it no longer has the ability to express its MHC1 protein as it should. It doesn't show that fingerprint. It doesn't have that signature that says, I'm healthy. It loses it. And as this natural killer cell is doing surveillance, it recognizes, here's a cell that doesn't have the signature that I'm looking for. And that stimulates it to activate, to bind and activate and cause the destruction of that cell, right? It eliminates these cells, hopefully before that virus has its ability to replicate and spread to other cells. We can target it and destroy it and maybe ward off this infection before it's out of control. Obviously, there's other issues, and we will be talking more about trying to deal with it, but the natural killer cells are a first line of surveillance to defend against some of these circumstances. So as we see that cell being kind of blown apart in that cartoon, one of the mechanisms that the natural killer cells utilize is a protein called perforin. It perforates, as perforin should imply. It pokes a bunch of little holes into the cell, and it causes it to lyse, right? Our own cells, right, that are virally infected. These are what causes the destruction. So this is a bunch of natural killer cells, and the granules you can see are, some of them, perforin proteins within there. So this image are the monocytes and macrophages. Just depends on the circumstances. So the monocytes within the circulation, you can see a bunch of them. Here's a neutrophil. This guy right here, lymphocyte. It's huge nucleus, very little cytoplasm. B or T cell, can't tell you, all right, but it's one of them, all right. And then a boatload of monocytes, right, right throughout, all right, a bunch of them. So in the circulation, they exist as monocytes. They truly want to leave the circulation and make their way into the tissues. That's when they become macrophages. And there are all sorts of different potentials with that. As far as body sites, different tissues, they're still macrophages, but we oftentimes give them different names. So like within the brain, microglial cells, Kupffer cells in the liver, ideas like that. There's a table in the text. I'm not going to switch out of that to show the table, but it basically lists off some of the differentiated macrophages. You don't need to worry about that. Just recognize in different body sites, they may have different names, but still macrophages. They're doing surveillance within the tissues, and they're really the first responders when we think about something with, in the tissue, a cut or a wound, something like that, that might allow foreign invaders to get in. Macrophages typically are what's going to be the first ones to see that and to stimulate a response. And they, can kind of move around by sort of amoeboid-like movement. I said I wasn't going to switch to the book, but that figure is pretty difficult to see. Let me stop this. See if I can get back to the textbook. Okay. Bringing it down. Here's the table. I said I wasn't going to show you, but I've already got it, so we may as well. So the microglial cells in the brain and central nervous, Kupffer cells in the liver, alveolar macrophages in the lungs, and then the peritoneal, within the peritoneal cavity are a few examples. There's also just straight up macrophages, whatever. All right. This is a little better to see. So with this, here we've got some sort of a wound, all right? So the skin and the wound, the green guys are the pathogens getting in, and we can see a few things happening, all right? So try to not move that out of the screen. So within the capillaries over here, we've got our red cells, we've got our white cells, our little blue guys right in here are the platelets. So we've got neutrophils and monocytes circulating through. We've got our capillary epithelial cells, and we've got the potential for these guys through diapedesis to move through these epithelial cells out of the circulation and into the tissues, right? Oh, maybe these guys were complement proteins because that's C5a, that's complement that's moving out. Whatever. So initially, as we start here, there's leukocytes responding to chemical attractants that are being released out. Potentially this macrophage sees these guys, whatever sort of pathogen it is, and it initiates this response. It phagocytizes, so it's starting, you can see right here, starting to engulf this bacterium. And in the process of finding these bad guys, it starts releasing cytokines, chemoattractants that tell, as they are released out, they tell cells that are nearby, we got a bad guy, whatever it might be. So this stimulates the cells within the circulation to start moving out of the circulation into the tissues and trying to fight off these foreign invaders. So the neutrophils, as they move their way out, they're ready to start phagocytizing, potentially also dumping some of their enzymes and stimulating more of a response that way. The monocytes, as they move out, will differentiate, basically turn in to more macrophages to try to help mop it up, if you will. So they squeeze through there, attracted by those signals, trying to recognize these foreign invaders and come in and deal with it. The neutrophil here, you can see releasing some of its chemicals, cytotoxic chemicals that will extracellularly kill some of these bacterial cells. As this neutrophil is doing this, as it's dumping these contents, it's taken one for the team as well. And it's not going to be able to do anything else. It's going to die in the process of releasing these chemicals. Same sorts of ideas if they're phagocytizing. So more macrophages mopping stuff up. You can see maybe, I think another image shows neutrophils phagocytizing as well, where they'll pull in. So within these tissues, you can see them releasing chemicals, destroying the pathogens, stimulating other cells, macrophages and other neutrophils to come in to recognize and respond. That's kind of the idea with that. So I'll go back to the text, I think. PowerPoint. All right. So these guys are phagocytic cells. So thinking like the macrophages, right? Neutrophils also have these potentials as well, all right? But phagocytic cells, those that are on these kind of seek and destroy types of missions, if you will, have specific receptors on their surface that allow them to really do the type of surveillance that's needed to recognize the foreign pathogens, whatever they might be. And there's a handful of different things that they're trying to detect, right? So these cells have these PRRs, pattern recognition receptors, and they're proteins that allow them to detect the foreign stuff, right? And what they're really looking for are PAMPs, pathogen-associated molecular patterns. So they're looking at signatures, fingerprints that are from a microbe and not something that's from our own cells, right? Not something, you know, our MHC1s are saying, this is me, leave me alone, right? And these cells see that as our own stuff, all right? But other cells, well, they don't have MHC1s. They have something else going on, right? Something else on their outside saying really who they are, all right? Their molecular fingerprint, if you will. So there's a handful of different things we might be able to pick up or to detect. Lipopeptides, so lipid and protein or amino acid fragments. Flagellin, numerous bacteria with flagella and specific proteins from that flagella that allow them to recognize that. And then peptidoglycan receptors, recognizing cell wall material of those bacteria. Three different potential mechanisms and some other potentials as well, thinking about maybe virus, where we can see other possibilities, but it's this sort of an idea, right? So there's more going on in this figure. With these specific receptors on the surface of this phagocytic cell, if we come in contact with some foreign invader, it will link up on this receptor and it stimulates this cell. Basically, it recognizes that we've bound something. It stimulates the cell to deal with it. Once we make contact with this receptor, with whatever it is, all right, we come in contact with the receptor and it causes an invagination. It causes the cell to basically engulf or surround whatever we've docked with, whatever we've linked up and snagged. So here you can see a bit where we've pulled it to the inside. right? And so once we've done that, we've got a membrane-bound vesicle, a phagocytome. This phagocytome has whatever we've pulled in. It could be the entire bacterium, right? Or whatever it is, right? Um, so that in turn can stimulate the cell to start releasing substances. We typically, and we're not showing all of the details of phagocytosis right at this point, but we will. That phagocytome will link up with a lysosome, becoming a phagolysosome, basically dumping from the lysosome, dumping those digestive enzymes on this foreign invader and destroying it with the potential to start presenting to other cells, as we may show fragments of that on the outside. It's not where we're at with this figure, but that's kind of the end result. So we can, as in this case, maybe this receptor detecting specific nucleic acid that is different and it stimulates this. So maybe we've detected some viral nucleic acids as this virus has tried to dock with the cell. We've pulled it in, and now we've told the cell, this is what we've got, and it starts releasing interferons that are antiviral, and maybe some other cytokines telling other cells to come in and respond. So this more detail of what I basically was kind of hitting at with the fusion of those lysosomes. So in this case, our bacterium has been engulfed. So specific receptor on that cell, maybe for peptidoglycan, has caused the cell to recognize I found a bacterium and it's pulled it in. It's within a vesicle, so part of the plasma membrane of the cell surrounds this bacterium. So again, we call that a phagocytome. So it's within a vesicle. The lysosome right here has the digestive enzymes. You see these guys fusing together to create that phagolysosome. And those digestive contents get dumped onto that organism within that vesicle, and it gets destroyed. So some of these particles get thrown out, like the trash, the undigested wastes get disposed of that way. There are lots of things happening inside of this macrophage where once we've destroyed, if we're truly talking about some foreign substance, so foreign invader, there will be stuff that's undigested and discarded, but there will be lots of things that are potentially important. Fragments of this cell that serve as recognition molecules that it will show to other cells. And this macrophage, as it's found these bad guys, will turn into an antigen presenting cell when it shows some of these bits on the outside, right? And that's in a different type of MHC molecule or protein. That's the kind of show and tell molecule that allows the immune response to be recognized. This image is a little bit disturbing, I think. One good picture to say, thankfully, we're in the middle of nowhere, Idaho, where you don't see any of this stuff happening, all right? That's good. So cutaneous leishmaniasis is a relatively tropical type of a disease, we will say. Not necessarily tropical. It's transmitted by a sandfly. So there are definitely issues of potentially seeing some of this in like Middle East countries, but definitely it's more common in warmer types of environments. We don't see it around here, right? And there's a handful of different Leishmania parasites. They can cause cutaneous issues. They can cause visceral issues, all transmitted by a sandfly, but different types of species within the genus Leishmania, different kind of manifestations. So here we've got some cutaneous involvement. We've got all sorts of kind of issues on the skin, likely all of these guys harboring some of the parasite. And this, I think, is kind of awesome. It's also kind of disturbing. In this part of the life cycle of this, Macrophages' jobs are to destroy bad guys. The macrophage engulfs these parasites, uh, but it doesn't kill them. They reside inside of the macrophage. They replicate inside of that macrophage. The cell that should kill them, they live in and they destroy, right? One of the best places to go undetected from the immune response is to take up residence within that stinking cell. If you're living in the macrophage and replicating inside of it, it's a brilliant strategy to evade destruction. The cell that should kill you is where you're living, and you destroy it in the process. So these amastigotes are uh, a stage within us that we see. They're kind of a circular sort of parasitic stage. Um, and that's what gets into these macrophages, replicates, releasing more of them out. They get mopped up by more macrophages that in turn allows them to just live inside of them. So all sorts of different possibilities with that. Real quick, we'll hit this. I think we're nearing the end. Mast cells, also important when we think about injury, and this with an inflammatory response. So they start releasing histamines. The histamines modulating this inflammatory response. You can see this capillary here and over here, it is dilated. So we've got increased circulation, increased blood flow, and the potential for really calling the troops, if you will. It stimulates a greater likelihood of these white cells showing up to this issue in the first place. So the histamine stimulating this, and again, inflammation can be good and it can be bad. And in instances like this, it's important to get these cells here and to try to deal with stuff. And that's really what we're looking at with that. Here's a granuloma. This is a tubercle granuloma in lung tissue with individual with tuberculosis. It's an immune response where we try to wall off the mycobacterium, the type of bacteria. And there are other diseases where we can see granulomas occurring as well, but it's an immune response trying to keep these guys in check or sequestered away. That organism is not killed within these granulomas. There are potentials that it can be released and let that parasite back out, and we can see issues with that. Elephantiasis, there's a handful of different types of filarial parasites that can get in, impacting the lymphatic system. We will hit some of these diseases in the next unit of material. But they cause blockages with the lymphatic fluid, and it's basically the fluid, the lymphatic system should be draining interstitial fluid. It should be moving this stuff out of the extremities. But as the channels get blocked, the fluid can't be removed out and it accumulates. So you can see all sorts of issues with this. And typically, legs, sometimes arms are impacted with it as well. Kind of depends on the specific type of worm. It is a type of roundworm or nematode that causes filariasis. And lastly, the hypothalamus is important when we think about inflammation and fever production, as we see specific substances that start to stimulate cytokines, stimulating cells to come in and respond. We see inflammatory process, and we also see these cytokines impacting the hypothalamus, so causing an increase in the temperature. Sometimes when we think about infection, the temperature, just a subtle shift in temperature, becomes inhibitory to some of these pathogens. So a little bit of fever might be a good indication that your body's trying to ward off this infection. High fevers can definitely cause some serious issues, and you may want to try to bring that down. So pyrogens are, a pyrogen is a fever inducer, or turns that thermostat up, if you will. Exogenous pyrogens, like the lipopolysaccharide layer from our gram-negative cells, are white blood cells. These leukocytes can release endogenous pyrogens. Different interleukins and interferons can deal with that. So stimulating the prostaglandin that basically cranks up that thermostat. All right, that's the end of that. Any questions or concerns? Okay, guys, I hope you all have a great weekend. Like I said, I will have some practice questions posted, hopefully by the end of today, hopefully sooner than that. That's my plan. Okay.